into groups of 2, and you see that everybody got the same diet, so there was a single intervention, which is the contrast in a trial. And the only intervention that worked was number 5, 2 oranges and 1 lemon given to each of the 2 sailors each day. There was a planning problem. He ran out of oranges and lemons in 5 days, but he was a lucky man. That was enough vitamin C to cure the scurvy. And he went on, wrote a thesis at Edinburgh, and became a professor. So there's an element of luck in everything. And as a professor, he had many theories about what happened, and they were all completely wrong. But his trial was transformational. The British Army and Navy had fresh vegetables and fresh fruit from then on. But his thinking about what the key factor was, the interpretation, needed a lot more help. So here's a modern clinical trial, and these are some of the tools that one needs. Clinical coordination of the doctors, patients, nurses, healthcare professionals, data coordination, people who manage the data. You need a good study design, and you need to train all the people involved in whatever is the intervention that is disruptive to the regular routine care. And you often train and retrain those people. It's more complicated than meets this simple eye. Here's a project management process. I'm not going to talk about each of the large list of things that we go through to come up with a plan. But there's a planning or setup stage, there's an execution stage, and there's a closure stage. And in each one, there's a set of 5 to 10 activities that you need. What we found, and what I mentioned earlier, we were lucky about, is this electronic data capture. We started in an age where there was a leapfrog. It's like you didn't have to have good landlines for telephone because you had mobile. The leapfrog here was to use the cloud as a central source to gather the data from all the institutions, the patients, and the physicians and the nurses. And that's what I want to talk about. We were lucky enough to have a system that was required by the FDA in that leapfrog situation that allowed for attribution of each entry from anywhere in the trial consortium by any person, so that we could track the activity of the people in the trial. I mostly do phase 2 trials and large phase 3 trials. A large phase 3 trial may have 1,000 people entering data. A good size phase 2 may have 200, 300 people. It's a very important tracking process. And we decided the FDA requires you control it so that they're certain that the data curation has integrity, that the actual data that's put into the electronic data system correctly represents the patient and then eventually represents the risk-benefit balance. We've turned that around to actually look at the activities that people are doing and help them know when they're not doing the right activities. And that's part of the story I'll tell you. So here's one can be overwhelmed by all the things that are needed to do in a clinical trial. So this is my diagram of what are really the 5 things you have to do. Screen and find patients, randomize them, do the intervention, and part of the intervention has 3 important subparts. You have to do the intervention the right way. Not the way you think it should be done, but the way it's described so that you and Zal are doing it the same way in different hospitals. Otherwise, you're not testing the hypothesis. You have to collect data on all the other treatments that the patient receives, and you have to collect data about important safety events. So those three things are part of the intervention. You then have to follow up the patient, and you have to do an analysis, and the analysis has to take, you can't cover anything up. It has to take into consideration what data is missing, and where you didn't do the protocol the way Zal told you to do it. Okay. When you have all that information organized, it turns into what's called a consort diagram, which is in every major New England Journal, Lancet, smaller journal publications, which show the flow of patients who received what and what group you analyzed. This is what people think of as clinical trials. In order to do those steps with all the minor steps, it's a big roadblock. How do you get around roadblocks? Well, with a lot of money, and that's what pharma does. So when pharma does a phase 2 trial, they'll put 300, 400 people on monitoring that set of events. I started out with 3 people. We've grown. We now do, currently we're doing 27 trials, but I do it with 70 people. So that's sort of 2.5 per trial. You have to know what's happening and understand how that bigger group of people who are putting their data in are doing. This is a diagram from the Pharma Association in the US, which is an international association, of the funnel from drug discovery, preclinical screening, clinical trials, to something that actually makes it to people. And this is why folks think a lot about the roadblock in trials. From an idea to actually changing practice is a long time, and most of the ideas are put aside, which in the end is a good thing if they're put aside correctly with the right decision making. Here's some demographics on funding through 2019. You can see that up here, the blue line, the funding is increasing for clinical trials, but the number of clinical trials is not really increasing. So they're becoming even more costly. And one has to consider how you allocate resources. So again, digitalization that allows a smaller number of people to manage bigger projects is one way to work on that process. Here's the results of a survey on that funding activity for clinical trials. Tasks are going up. The frequency with which you do a task of data collection is going up. The work effort to do that is going up. And sometimes this is because you use multiple platforms within your healthcare system that require interpretation, and then you reload data into a system. It's not so good. And I guess that's execution burden. And payment isn't changing, as you can see. So the first thing to understand is everybody has trouble with trials. And the solution is not digitalization completely. The solution is people who use digitalization. These are the 4 main people who work with me, who've done multiple clinical trials, and help an investigator, like Sandor, navigate all of these tasks with digitalized tools. So how do we start a trial? We have to build a consortium and find like-minded people who want to do Sandor's trial. So we start with a web-based survey. We call it SASE. And the idea is we have an effective trial design, we have some statistical planning, and we need to find across our country or across the world a set of investigators who are willing to disrupt their care and do the intervention. This is our network across multiple universities that doesn't require the rector or the department chef to say, yes, you can do this. They can just come to us and say, we have an idea. Only 1 in 5 gets to the trial stage, but they can access this capability. And we survey them through this network. This is what, on the left, a network for a trial is going to look like. And on the right, it's an example of all the human interactions that go on at each of the sites in order to meet the patient, have the patient agree to the trial, and then gather the patient's data from a lot of different areas. So quite complex. And we use the survey with the plan to see how well or how poorly the site is interested in the trial. So when we survey the study PI, the team of healthcare professionals that support them, and they interact with us as the coordinating center over all of the purple areas. And we can now do this. We've done it about 15 times, and it takes about 2 hours of time for each site to complete the survey. And in the end, sites are happy if they choose not to do it, and they're happy if they choose to do it because we have shown them what they need to do at the very beginning, and we've learned who the decision makers are at that site. About 70% of trials experience startup delays because, let's say we're dealing with 30 sites. We don't know all the important people who must sign off to the university at each site. But with this instrument, we've gathered that information, and we've gathered it just in time for the start of the trial. Approximately 4 out of 5 sites fail to meet their enrollment goals. But with this instrument, we've asked them to interrogate their own internal electronic system and see if the patients are there, and they've begun to think about that. So this beginning of a planning stage, we believe, allows for earlier completion, more reliable data, at lower cost and less burden to both the coordinator and the coordinated. The key way we manage information is the electronic data capture. A single cloud-based system that is designed specifically for the data needed to answer Sandor's hypothesis. And that either the 200 people in a phase 2 trial or the 1,000 people, each with a unique credential, each with unique permissions. So if Sandor is studying Zal's drug, Zal cannot see the results. And Sandor can't see the results of grouped patients, only the results of his patients. So the permissions allow for appropriate blinding and management of knowledge to produce objectivity. On the left, you see the 3 stages again of a trial and how one uses the data from the electronic data capture system. Here's a schematic. I'm not going to talk about this trial. This trial is one where we started last year, which uses the Apple Watch to report on how the patient is doing to this electronic data capture system. And through a platform called Eureka in the lower right, takes that data and then feeds it back to the patient as actions the patient might want to take. But this is the benefit of the cloud. So we picked our sites. The sites are willing. We've done study planning on the left. And now we have to start the regulatory process where each site, Tbilisi State University, must conform to regulatory and training activities for the study and for Tbilisi State University. They must get IRB approval for ethical approval, and they have to do contracting. And all of those things have to occur before the site can begin to enroll patients to take Sandor's intervention. This process can take up to a year. We streamlined the process into steps that were easy to digest over, if you look at the top here, a 12-week period of a curriculum. And these are all the steps that are required. And we have the person at the site enter what they're doing, and the person from the coordinating center can tell them if they're on time. If they're having trouble, the person from the coordinating center can see they're having trouble. They call them up and say, how can I help you? With that process, we tested the hypothesis that we could take the start time, my start time from the MISTY trial, which I've shown here, 275 days, almost a year to get a site on board, mean, okay? Down to 90 days. This is what we did. You see, during the pandemic, for a couple of facility availability reasons in outpatient trials, the times were longer. But with a simple curriculum enforced by a human on the phone and a cloud-based system showing the progress at each of the 30 sites, we bring it down to 90 days. You ask the question, who is working for who? And is this a very difficult job? So we try to make the job more fun. And we take those tasks and we made it into a game, climbing Mount Everest. And at each stage, you got feedback as to how well you were doing and how well you were doing compared to the standard. We did not compare you right away to your peers, just to what we asked to do. And if you look at the lower part, these are the survey of the people who did the tasks. And when they did the tasks playing the game, where we showed them, again, using the electronic database to show them how they're climbing Mount Everest, they enjoyed that, they felt good about it, and they were engaged, and they did the task in a better way. So this is the theme I'm showing you. Prelude is the electronic data system that we use most commonly. We also use REDCap. You have a data export process that is managed by people who are only managing that, who aren't doing the intervention and aren't doing the assessment. It goes to our local warehouse, and then we build reports on the progress. And here's an example of the screening and enrollment stage of how well a trial is doing in screening, and what are the reasons for the patient not being enrolled, which again is a mitigatable event. And here is a monthly key performance indicator dashboard. And if you look at the top, we plan ahead on the top part of KRI definitions and thresholds. What are the things that are likely to give us trouble, that have given us trouble before? And what's a threshold for performance at Sandor's clinic where we begin to say he might need some help? We do this and we look at it either weekly or monthly. All right. Let's move on to a true digitalization effort. My trials and my research are about brain hemorrhage. And early on, we were some of the first to demonstrate that how big the hemorrhage is matters on how likely you are to have a good or poor outcome. And we tested, the intervention was to reduce the size of the hemorrhage. So accurate measurement became important. We were doing this in a disconnected way. So the problem for digitalization and the opportunity for a digital transformation was to unify the information from every site to the coordinating center and to make the measurement of the size of the hemorrhage uniform and rapidly available. In the MISTI 3 trial, we did this with 4 computer systems, probably 6 human beings, many telephone calls, and transfer of information from one digital device to another. And also some phone calls sending pictures. I was lucky enough, and the National Institute of Health partnered with commercial businesses, 2, Google and Amazon, and they invited us to ask for help to develop cloud-based digitalization systems. And the benefit was we have a digital starting point, as you talked about yesterday, the CT scan. We had developed by hand a so-called heuristic model, a man-made model with man-derived measures of what the model does and how it works, to localize and quantify the hemorrhage size. And we then took that as a ground truth, as a gold standard, and used it for machine learning to develop a more rapid method that didn't require man. And this is that process, okay? On the left-hand side is the CT scan, the white is the brain hemorrhage. We built a user interface, so it goes directly to the cloud, the file just goes to the cloud. A human has to send it. In the iteration we're currently working on, it comes directly from the CT scanner and the PACS system, so no human touches it. It's de-identified so that the issue of personal information in the cloud is handled. Only Sandor at his site with a key can identify it. It's sorted, and then an algorithm produces what's on the right, segmentation of the clot into 2 major components, the interventricular and the blood tissue. This is the comparison. The hand is a human segmentation of the clot, and the T uses TensorFlow, which is a operating system that Google has in the cloud, and they've released the code for this so everybody can code their problems. The blue dots are when humans do the volume measurement task, and the red dots are when our algorithm does it. And the 2 lines, this is called a bland Altman plot, which is a measure of error. We felt it was acceptable to have plus or minus 5 milliliters error. And the algorithm operated on in the cloud, built by humans, backed with a gold standard, performs quite well. This led to a publication in Journal of Neuroimaging, in which we validated this in a large open data set. And you can see in the blue box how good the measurement is. We now can, instead of 5 hours, when Sandor's CT scan arrives, he gets back this segmented result in 100 seconds with more accuracy than his best fellow can generate. So how can this work? When we started the COVID pandemic, colleagues came to me and said, we're tracking it internationally at the Johns Hopkins School of Public Health. We want to test if the old treatment, which has 5 Nobel Prizes associated with it, antibodies, could help in the early stages of COVID. And so this is the trial that we designed. You draw plasma with antibodies from the COVID person who has recovered. You only use the highest titered people so that you're giving to the new COVID infected person a very high level of antibody. And we randomized. We gave a single dose, placebo to half, this is the orange column, and the convalescent plasma to half, and we showed a 50% reduction of need for a hospitalization and all the other concurrent symptom reductions. And we were able to do this using the system I showed you in 15 months, organizing 30 sites to do the same thing at each of their sites. And it led in month 16 to the changing of the FDA allowance for this drug. And we're now in the process of negotiating a permanent allowance with the data we collected on this electronic data capture. And it led to this publication in the New England Journal. Despite the fact, you will like this, Otto, we did this without the blessing of Dr. Tony Fauci. He said this won't work. I'm the professor, I know better. Just a modern example of why we need many people doing things with good tools and good intention. So, what did we learn? Each trial requires a leader. It requires a Sandor to advocate for the intervention. It requires a consortium of people who will disrupt their lives and their patients' lives to do the intervention. And it requires an organization like I've just shown you. We now have a learning system with the EDC that we can interrogate about how long it takes us to do tasks and how well we can do that task or not. Implementation requires planning, lots of planning. And on the right is what I've told you. These are the 5 things you must do in a clinical trial.